Thanks, Andrew. And we are going to hold questions until all of the speakers have given their talk. So if you have any, hold on to that thought and we'll get back to you at the end. All right, so next up, Bryn Phillips is a research specialist with the University of California, Davis Department of Environmental Toxicity and has worked at the UC Davis Granite Canyon Lab since 1992. During his career, he has managed aspects of a number of large-scale monitoring projects that most recently include the Surface Water Ambient Monitoring Program, or SWAMP, the Stream Pollution Trends Program, SPOT. Mr. Phillips has conducted a number of research projects related to ag and urban runoff, as well as research projects funded through agencies such as the California Water Boards, Department of Pesticide Regulation, and various resource conservation districts. Please welcome Mr. Phillips.
This goes back, that goes forward.
Awesome. Mr. Phillips. I'm not a data scientist. I wish I was, though. I might get paid more money. Today I'm going to kind of go out on a limb and talk about something that I'm not really super experienced in. I dabble in policy, but I'm not a policy person. I'm an academic researcher. But in doing the research and monitoring that we've done at the Granite Canyon Lab, we've broached policy topics related to pesticide regulation and TMDLs and sort of beating the drum for changes in types of monitoring and getting ahead of processes. And so I find myself here at the behest of my colleague Brian Anderson giving this talk, talking about pesticides of emerging concern, current use pesticides, and TMDLs, among other things. So there's probably a lot of people in this room that know more about aspects of what I'm going to talk about today than I do. So I welcome, during the panel discussion, corrections, comments, illuminations, anything you want to give me. I basically want to talk about three topics today, and the first one is the pesticide cycle. And have people heard this term before? Is it kind of making its way through the world? The pesticide cycle is a term that I like to think that, you know, our circle of colleagues has coined, but it's probably been around for a while. And with Deborah Denton's permission, I'm posting a couple of figures and tables from her upcoming paper, which will be submitted to Environmental Toxicology and Chemistry, that talks about sort of getting ahead of the pesticide cycle. So the pesticide cycle, the way I think about it, is this succession of new active ingredients replacing old active ingredients in commonly used pesticides. So, you know, if you're a homeowner and you go and buy ant spray, if you bought it, you know, 20 years ago, it might have had organophosphates in it. If you bought it 10 years ago, it might have had pyrethroids in it. And if you're buying it today, it might have fipronil or a neonicotinoid in it. It's still ant spray. It's still Raid. You're still spraying it around your house to get rid of ants. Particularly, and I always like to quote my dad in Southern California, because he always has a bottle of bifenthrin currently sitting on his doorstep to kill all the ants in his around his Santa Monica home. He hates ants. But this succession of pesticides, we all know about it, and we're always trying to stay ahead of this succession of pesticides on the monitoring side of things, which is what I know most about. And traditionally, different organisms that are commonly used in toxicity tests, which is what I do day to day, are more sensitive to these certain chemicals than other groups. And so back in the day with organochlorines, fish would bioaccumulate these chemicals, and they seem to be the most sensitive to the organochlorine pesticides. As time went on, we identified organophosphate pesticides in ambient waters using Cereodaphnia. Cereodaphnia is one of the three sort of benchmark species that are used in EPA testing. We call it three species testing. It's Cereodaphnia, which is an invertebrate, fatted minnow for the fish, and Selenastrum for an alga. And none of these three species were sexy enough to get on the sticker today, I noticed. So I'd like to talk to whoever made this thing, because the the Ceredaphnia is sort of like this cornerstone species for toxicity testing. So I'll bang the drum for tox testing. Moving forward, though, using Hyalella azteca and amphipod in sediment testing, we identified a lot of problems, water quality problems with pyrethroids in sediments. I'm not going to get into the intricacies of how we sample sediments, but I'd like to talk to you a little bit later about how we can improve things. And then moving forward, Chironomus dilutus, which is a midge. So we've gone from a cladoceran with Ceredaphnia, Hyalellas, and amphipod, and now we're working with a midge. And all of these species are different, or have different sensitivities to the various classes of pesticides. And so I have some numbers to back these claims up. And these are 96-hour median lethal concentrations. And this is the amount of a pesticide that'll kill half of the animals that you expose it to. And so for chlorpyrifos, if you look at I don't know if I have a laser pointer here, but no. Not much of a laser pointer. But yeah, so the shaded boxes there illustrate the most sensitive species. So chlorpyrifos, Ceredaphnia has the lowest number, so it's the most sensitive species. And then you can see Hyalella azteca is sensitive to bifenthrin. Chironomus sensitive to fipronil, which is currently an urban use pesticide. And then imidacloprid, one of the newest ones, it's a neonicotinoid, sort of a toss-up between Ceredaphnia and Chironomus. So we've talked about pesticidal and organism sensitivity. Those are the two main topics. And I'm going to kind of beat this drum about organism sensitivity for a little bit. Because in the monitoring work that I've been doing since 2001 for swamp, when we analyzed the first 10 years of data, we found that greater than 50% of our collection sites had some form of toxicity at that site. And it's not 50% of the times that we collected a sample, but it's 50% of the sites. So a lot of these sites are visited multiple times, and if they have one measurement of toxicity during the times we visited, we counted it towards this 50%. But that's a lot of toxicity. And we found that most of the toxicity was associated with pesticides. And then more recently, for the Stream Pollution Trends Program, SPOT, which has been going on since 2008, we have 1 in 5 sediment samples significantly toxic every year, and toxicity has the strongest correlation with urban pesticides. And in our data sets through 2014, most of the urban pesticides are pyrethroids. Again, see my dad sitting on his doorstep spraying ants and then watering his lawn. That's one of the main transport mechanisms for these chemicals getting into the water bodies. In the SPOT data set, since 2008, median concentrations of pyrethroids have more than doubled. And in the years we've been measuring fipronil, which is only 2013 to 2015, we've also seen median concentrations double. I know that's a short time frame for a trend analysis, but it is anecdotal. So just a couple of maps. I don't know how well everybody can see these, but on the left is the 6-year average magnitude of toxicity, where we're averaging our SPOT data set to look at how much toxicity is at each site. We visit these sites once per year, so it's a trend project. And I've juxtaposed that against the 6-year average organic carbon-corrected pyrethroid toxic unit. So this is a way we measure pyrethroid pesticides. We sum them, and then we convert them to toxic units. So this is how sensitive the organism is to that pesticide. And you can see that the circles, the big circles, sort of line up with one another. The big circles of toxicity line up with the big circles of pesticide. And so we see this pretty significant relationship between the two. If you look at a fipronil map, again, this is only 3 years of data. This is only at urban sites. And it's the same sort of thing, particularly in the Southern California regions. So you have detections of toxicity, and you have detections of these chemicals, and that generally leads to water body listings and eventually TMDLs, total maximum daily loads. So what I've done here is listed some local TMDLs. This is from Region 3, the Central Coast region, and these are TMDLs that are related to chlorpyrifos. At least the first 5 are related to chlorpyrifos. There's a Santa Maria watershed TMDL that's related to pesticides, and then in development there's a lower Salinas River watershed that's focusing on sediment toxicity and pyrethroids. The interesting thing about these TMDLs is the data that went into them, and Karen Worster's sitting right up here from Region 3, so she can correct me if I'm wrong on any of this. The data that went into them was collected as early as the early 2000s, and all these data points go into create this water body listing and create this TMDL. These TMDLs are implemented, as you can see from the dates, from 2011 to 2014, and they have attainment dates of 2016 to 2025. So I think that's a pretty long process for identifying chlorpyrifos as an issue and then actually having a complete reduction in a load and sort of terminating the TMDL. And we know over the time that these TMDLs are running, we're already seeing changes in the pesticide use. So I have a couple papers here from 2006 in the Santa Maria watershed where we were already identifying pyrethroids as the cause of toxicity, and in water column and in sediment. When we did our urban pyrethroid project under swamp, we found a number of sites that had pyrethroids contributing to observed sediment toxicity. This is around 2010, 2013, I think this is, or 14. More pyrethroids with some OPs in Santa Maria watershed. And most recently, we did a study on Quail Creek, which is a tributary to the Salinas River, which was traditionally very, very toxic and toxic to Daphnia and was full of chlorpyrifos. There was only one detection of chlorpyrifos in all of the surveys we did. We were actually treating this creek with an enzyme to treat chlorpyrifos, and we didn't have any chlorpyrifos in our creek. So it was a bit of a conundrum, but we did see pyrethroids in the creek, and we saw plenty of Hyalella toxicity, so it's a good thing we added that extra species to that study so we could we could notice that change in pesticide use and in the change in the concentrations of the pesticides in the water body. Most recently, Brian Anderson has written a paper, this is in Press, and it's changing patterns in toxicity associated with current use pesticides. This is data that's based on a study we did with the Department of Pesticide Regulation, where the water boards at Region 3 and Region 7 funded us to do toxicity testing and do some pesticide analysis at a number of their ag sites. And we were able to compare those two regions. And you can see that as a consequence of the discharge requirements, there's a shift in pesticide use patterns. So Region 3 has more policies that focus on chlorpyrifos, and as a result, you see less chlorpyrifos use in that region and less chlorpyrifos in the water bodies. And the toxicity is different also. The toxicity that you see in these water bodies is now not related to Daphnia anymore, which is one of the 3 species that are used for testing. It's related to Hyalella and to Chironomus, which are now becoming species that are being used as a monitoring, in the monitoring process. I like to think that's a result of us harping on the various water boards for the last couple of years, and hopefully this paper will kind of put a lot of arguments to rest on organism sensitivity. So the third thing I'm talking about is proactive strategies, and this is where I get into unknown areas. I've already talked about Deborah Denton's paper, which is in prep. And then I've already talked about Brian Anderson's paper, which is in Press. And then there's also a third document that I reviewed among various other websites. That's from the Strategy to Optimize Resource Management of Stormwater, the STORMS program. And one of the objectives of the STORMS program is to reduce urban pesticides. And all 3 of these sort of headings all have similar recommendations, similar strategies to try to get ahead of the pesticide cycle. So we have these water quality issues that we identify. We can relate them to pesticides. We respond with a TMDL process, but I think all of these groups want to try to stop the problems from happening in the first place. and try to figure out a way to have an alternative to a TMDL. And so the number one thing that Deborah and Brian and the storms groups all talk about is having agency cooperation and coordination. And I'm familiar with the management agency agreement. I know my colleague DeWeet sits on that group. And it's an agreement between the State Water Board and the Department of Pesticide Regulation to try to keep communication lines open, share data, become transparent, and know what each agency is doing to try to get ahead of this problem. There's two agencies that regulate pesticides. There's DPR in California, but there's also the U.S. EPA Office of Pesticide Programs. And if those two agencies are speaking to one another nationally, locally, and nationally, I think we can get a little bit closer on the registration side of things to prevent some of these problems from happening. DPR also has a prioritization model. And so one of the problems we're always having and asking people, we ask our colleagues at DPR and elsewhere, what is the next pesticide we need to be worried about? What do I need to be monitoring for? What's the organism I need to use for sensitivity? And how am I going to know this? And DPR has recently developed a pretty slick prioritization model that incorporates pesticide use, chemical properties, monitoring results, application information, and tries to give us some indication of what the most important pesticide is to pay attention to. I mean, resources are limited, pesticides are plentiful, so we have to decide which ones we need to focus on. And the results of the model can inform us on which analytes to look for, what kind of organisms to use, what kind of tools to use, and hopefully it helps us get ahead of the pest cycle and instead of being reactive. So I've talked about using other organisms in monitoring as well. I mean, right now, monitoring in terms of the regulatory world, you know, we monitor with toxicity and we monitor based on dead organisms, and we have concentrations of these chemicals that are high enough to kill these organisms that we use in our laboratory tests. SPOT has used Hyalella azteca since 2008, but recently we've introduced Chironomus in our sediment tests to try to look at fipronil toxicity, potential fipronil toxicity, because we know that fipronil use is on the rise and detections are on the rise. So what I did is I put the Hyalella data and the Chironomus data from 2015 and 2016 side by side just to sort of demonstrate that the toxicity with both organisms is pretty similar. The two stacked bars on the right, for Chironomus, you actually sort of see an increase in toxicity, an increase in those darker colors at the top between 2015 and 2016. So there might be a trend going on there. But the main point of the slide is to tell you that the sites that are toxic to Chironomus aren't necessarily toxic to Hyalella. There's a few sites where we've picked up new toxicity with the chironomids that we weren't picking up with the Hyalella, and that's likely due to different sets of analytes that are in the sediments. Once I get the 2016 data, I'll be able to tell you more about that, but for right now, we know that the organisms are telling us that something's going on with different sets of analytes. We want to sort of take this one step further. In 2018, we're going to be working with DPR again to try to do water column testing, and we're going to get funding for water column testing at DPR's surface water sites so we can add Chironomus and Hyalella to try to get at whether or not there's significant concentrations of these chemicals causing toxicity. So we're not just thinking about toxicity, and I know Alvina will be happy to see this slide. She'll be talking later about in vitro bioassays, but we are trying to pay attention to the latest tools that are coming about. And in vitro bioassays are cell lines that respond to various toxicants. There's a potential for high throughput with these tests and low costs. And we're hoping that there can be some versions developed for neurotoxic modes of action The insecticides that we're concerned about affect the organisms with a neurotoxic MOA. So if we have neurotoxic in vitro bioassays, that would be great, because it'll help us link the results to the organism responses that we see in our sediment tests and our water tests, and for potential community effects. So the last thing is management practices. The whole idea is to try to keep the pesticides, on the field, on the property. There's been regulations and label changes that have been implemented to try to make this happen. But inevitably, we think that pesticides are going to move off of properties and try to get into waterways, and we want to treat the water whenever possible to prevent that from happening. And so the photo on the left is a bioswale, an integrated treatment system on a test farm in Salinas that uses compost installations and grassline ditches and a granulated activated carbon treatment at the end to remove up to 98% of the pesticides that run through it. And then on the right is an established bioswale that's at a Home Depot parking lot in Salinas. And we did some monitoring on this bioswale during some stormwater work and found that it also removed a tremendous amount of not only pesticides that are applied at this complex, but also hydrocarbons, metals, and pretty much everything we measured. So to wrap it up, the idea behind this talk was to try to just present some strategies to create alternatives to TMDLs. I'm not an expert on the policy side of it. I'm a lab rat. I do toxicity tests and get muddy in the field. But I'm hoping that that everybody's sort of jogging their memories and thinking about ways of making some changes in the future. We want to become more proactive than reactive, and I've been told to stop, so I'm just going to let you read that last bullet. The strategies are the 4 main bullets that were pretty much put out in all of the documents that I present. All right, thanks.